Oh hi. Didn't see you there. I was busy looking for different types of microbes. But it is not that easy to see them. While we are made up of millions of cells, whereas microbes are just made up of one cell. They are so tiny that you'll actually need a microscope to see what they look like. Now, do you know where they live? Everywhere. They are present in the soil we play in, the water we drink, the air we breathe, the food we eat, and inside us too. In fact, they can live in the harshest conditions. They can live in cold mountains filled with snow. You can even find them in hot springs with high temperatures. They can also survive in deserts with harsh conditions. You'll be surprised to find them in deep oceans as well as areas with very harsh chemical conditions. And there are so many types. They could either be prokaryotic, which is basically like an ancestor. This includes categories like bacteria, which do not contain any nucleus or organelle. They can be seen in various shapes such as spherical, rods, helical. Then there are eukaryotes, and it has a variety of types like fungi, which is multicellular with filamentous hyphae, and yeast, which are unicellular and round. Algae, which contain chlorophyll and are photosynthetic. Protozoa are unicellular, free-living organisms that lack a cell wall. Microbes can exist anywhere as long as there is food they can survive on. Scientists like me grow microbes on special media that contain food for them. These are called growth media. Different microbes have different nutritional requirements. Let's take a look at what the ideal growth medium should be like. Let's add in the following. High carbohydrate source. Good nitrogen source. Yeast extract. We must also keep in mind that the temperature and pH levels are right for the microbe. Let's take a look at some cool microbial growths on different media. Now, as you know, there are so many species of microbes present literally everywhere. So, how will you be able to study them? To be able to study microorganisms, it is important to separate them from other species. We can, therefore, isolate microbes in pure culture via a process called isolation. Let's take 3 situations. First, let's isolate microbes from this soil sample. Take some soil and add it to a test tube containing 10 milliliters distilled water. Now comes the fun part. Serial dilution. As the name suggests, we will dilute our sample. In this we will take 1 milliliter of the test tube, then add it to the next test tube. Then we'll go 1 milliliter from this test tube to the next one, and so on. Can you see how the color is fading in the tubes? Want to know why? Well, you'll know soon. After we are done with serial dilution, we go to the plating part. Here we'll pipette out the sample from the test tubes onto the respective plates. Once the solution has been added to the Petri plate, we will spread the solution onto the plate using a triangular spreader. This is to ensure that the microbe-containing solution is evenly spread out. This process is called spread plating. Notice how the colonies are reducing according to the more diluted samples. This is because the more that the sample is diluted, the less will be its microbial population. If you want to isolate microbes from water, just filter it out, drip the membrane into broth, and then follow the same process. Finally, let's isolate some microbes from the air. One is by simply putting the Petri dish containing solid media out in the air for a little while, on which microbes along with the dust particles attach to it. The other way is by keeping the Petri dish with media in the automatic air sampler, by which dust particles from the air are attached to the plate by suction created by the air sampler. Look at these dots. They are so colorful and diverse. These are called colony-forming units, CFU. Each CFU contains one type of microbe. So our focus is to identify these microbes, whether they are bacteria, fungi, or yeast. So, in doing that, we first going to isolate the microbes from that mixed population. For this, we'll take a transfer loop that has been in contact with CFU and then streak it across the surface of agar medium. This is called streak plating method. The method gradually dilutes the CFU count with every streak. Now that we have a pure isolated culture, we need to give a name to what type of microbe has been purified. We must observe them and study their characteristics. It will tell us whether these microbes belong to bacteria, fungi, or yeast. Phenotypic characterization of microbes helps us to identify the microbes to their genes level. After colony morphological characterization, we go for microscopic observation to study the cell morphology characterization such as cell shape, size, motility. For this, we use a light microscope. Then comes Gram staining, which was developed by Hans Christian Gram, and it is used to differentiate between gram-positive and gram-negative bacteria. We will take our inoculum and smear it on the glass slide, then add our primary stain, which is crystal violet, followed by a mordant like Gram's iodine. Then we'll decolorize this by washing the smear with ethanol. Finally, we'll add in safranin for counterstaining. Now you could end up with two results. The cells which retain the crystal violet appears violet, called gram-positive bacteria, and the cells which loses crystal violet are counterstained by the safranin, and hence these cells appear pink in color, called gram-negative bacteria. Do you know why this happens? Because gram-negative and gram-positive differ in their peptidoglycan composition. Then to visualize yeast and fungi, we use a different stain called lactophenol cotton blue. First, we put a small piece of fungal or yeast growth on the slide and add lactophenol cotton blue and cover it with a cover slip. On microscopic observation, you will be able to see the hyphae of fungus and oval-shaped cells of yeast. Finally, our third method is biochemical and physiological characterization. Here, we figure out the different types of enzymes or byproducts produced by the microbes. Similarly, physiological characteristics such as pH, temperature, gas production, oxygen requirement, etc. The variety of these reactions help us to characterize and differentiate various groups of microbes. Our final step is to identify microorganisms, which can be fulfilled by molecular identification method. So the identification is done on the basis of its genetic material, that is DNA. Let's say that the colony you grew was one type of bacteria. Now, how will you know what its name is? Which genus and species does it belong to? This can be done by 16S rRNA gene sequencing. For this we first need to amplify its 16S rRNA genes, which are conserved sequence in bacteria. In species of fungi and yeasts, we amplify the 18S rRNA genes. This is done by a technique called PCR. We will first denature the DNA strands. This is done at 92 degrees Celsius, where the DNA strands start to unwind. Then comes the annealing stage at 54 degrees Celsius. Here the primers, which are short polynucleotides, attaches themselves to DNA strands. That can act as a starting code of DNA synthesis, and with the help of polymerase enzyme, new nucleotides will be added to the DNA strands. Finally, comes the extension stage, where polymerase enzyme add new nucleotides to both the strands to produce complementary strand. Thus we went from having 2 DNA strands to 1 billion copies in 30 cycles. How amplified DNA strands were subjected for Sanger sequencing, which is a chain termination method that helps in determining the nucleotide sequence of DNA. Sanger sequencing generates a chromatogram. This chromatogram is used to run a blast on the online nucleotide database such as NCBI. Here, our isolated 16S rRNA gene sequence compared with the sequences in the database to find the similarity between the nucleotide sequences. Based on this nucleotide similarities system, will generate a phylogenetic tree which shows how isolated microbes is evolved from a series of common ancestor. Now, we get to know what species we had cultured. This was just one of many microbes. Our world is filled with microbes, some good and some bad. Our friendly microbes can help us in so many segments of our life. For example, nitrogen-fixing bacteria like Rhizobium, they help in the growth of legumes via nitrogen fixation. Microbes like yeast, Aspergillus, and Lactobacillus can help in the food fermentation. The addition of the bacteria into milk turns the milk sample into curd. Using microbes such as fungus penicillium can be used to create antibiotics like penicillin, which can help cure diseases. In fact, we have a lot of friendly microbes are present in our intestine. It helps us absorb nutrients and fight off bad microbes. Take a look at the clothes you are wearing. Look at how vibrant the colors are. You know that microbes were used to ink your clothes? Microbes are being used to produce ethanol for biofuels, which is produced from lignocellulose, a mixture of cellulose, hemicellulose, and lignin, which make up the plant cell wall. Now, the question arises, are all microbes good for us? No, not all microbes are good. Some microbes can actually cause us a lot of damage. Like E. coli could make us sick. Or the Pythium species of fungi could cause diseases in plants. Or how fungi and bacteria cause food spoilage. However, we shouldn't let this scare us. Microbes are very ubiquitous and versatile in nature. They can be so fun to study about, and we can always learn so much from them. So, make sure that you observe nature around you and see what big changes these tiny friends can bring. So, here's till next time. Keep exploring.